This program is brought to you by Emory University.
Research is one of the most exciting things about science. In our biology courses at Oxford College, students get to investigate microbial communities of a local granite rock outcrop, Arabian mountain, like real research scientists, by thinking, doing, and communicating. This lab received the 2011 Inquiry-Based Instruction Prize, awarded by the journal Science. The first step of being a researcher is the thinking phase, writing a research proposal. The instructor partners with librarians to guide students in exploring scientific literature for this first step. Research teams then use this information to develop their own investigation questions and to make an experimental plan. Each research team must collect evidence, the doing phase, to support or falsify their hypothesis. The class arrives at the field site to collect bacteria samples. Each team selects their own collecting sites at Arabian Mountain to obtain soil or water samples. Samples are diluted in buffer and spread onto an agar plate. Plates are incubated for several days to stimulate bacterial growth. The next set of doing steps occurs in the laboratory. Students examine their plates to identify distinct types of bacteria from their sample sites by observing morphological characteristics of bacteria such as color and colony structures. Team members work together to select a set of distinct bacteria types for the next step. Before any conclusions can be made, student teams need to discover the genus and species identity of the bacteria that they have collected from Arabian Mountain. This is done by molecular analysis. DNA is extracted from each bacteria type, and the 16S ribosomal DNA sequence is amplified by PCR. In the next step, the DNA amplified by PCR is purified for sequencing analysis using a purification kit. Purified samples are sent to the Georgia Genomic Facility for sequencing. Samples of the same DNA, the 16S rDNA, are also prepared for a restriction enzyme digest. DNA digested with restriction enzymes are loaded onto a gel. Gel electrophoresis determines the extent of diversity among bacteria samples. The last step of the doing phase is where each team analyzes their DNA sequences that were sent back to them by the sequencing facility. DNA sequence data is entered into bioinformatics tools such as BLAST and Ribosomal Database Project. Teams are able to thus determine the taxonomical identity of each type of bacteria in their collection. After all the data is collected, research teams have to engage in another thinking phase of synthesizing information and evidence. Students discuss evidence and conclusions with each other. They consult with the instructor and make decisions about data presentation. Finally, it's time for the communication phase of the research process. Each team communicates the findings of their experiment at a class research symposium, presenting the evidence related to their research question, such as the different amounts of bacteria found in different locations or the different types of bacteria discovered in their samples. Then each individual student writes a paper describing their experiment to convey their own scientific thinking. This concludes the student's journey of investigating Arabian Mountain by thinking, doing, and communicating like a scientist.
The preceding program is copyrighted by Emory University.